Okay, I'm going to attempt to record doing this. I want to go over some of the nuances of this. So in this first section, they say to gather all your equipment. That I've done already. And the next thing they tell you to do is to crack an egg and then separate the yolks and then whisk the whites. And so I did that in a bowl because I didn't want to waste a plastic cup. So there it is. And I did use a whisk instead of a fork, but of course you can use a fork. The next thing they want you to do is to label a graduated pipette, a jumbo one. So there's different sizes. There are different sizes, and a jumbo one is this one. So you can see that it's graduated. It actually has graduations on the side of it. And so that's the one that you want to use for that. And so they say to label one of them, let's see, graduated pipette albumin, and then place it in the cup with the egg whites. They then say to label 5 test tubes with a list of different things. I did that, and they said to use the 24 well plate to put the tubes in. So I did that. I think it's just so it's easier to see the results. But I'm actually going to move my albumin stock tube to here. Okay, this is the little handy dandy test tube that they gave with the kit. Okay, so the next thing they tell you to do is to add 1/2 ml of egg whites from the cup or from the bowl into the test tube labeled egg white albumin stock. So I actually marked my pipette because it's actually graduated, but it's really hard to see, and with my old eyes. Anyway, so I marked it with my pen where 1/2 ml was. And so what they say is to put it into your tube that says the egg white stock. So there it is. Then they say to add 4.5 mils of distilled water using the graduated cylinder that it comes with. And so here's our graduated cylinder. And again, it's hard to measure because it's so small. What I had to do was I put a little bit of water into it, and then I used one of these graduated pipettes to kind of drop it in there to get 4.5 mils. One of these is going to be designated as your water pipette, so might as well use it now. Okay, so then I'm going to add this to my tube. Carefully. Oh, gee, I just spilt everywhere. I got to start over again. But you get the point. You get the message. I'm going to start over again. Okay, then they say to kind of swirl it around. But I think even a smarter way of doing that is to take your albumin pipette and then pipette up and down to kind of mix it up. It doesn't have to be exact. So just, don't get upset if this isn't perfect, okay? We're not asking for perfection here. So then they said to just leave the albumin pipette in there and for later. Okay, so the next thing they want us to do is to put 1 ml of the distilled water, 1 ml of albumin, and then 1 ml of milk into those tubes. And they want us to label new pipettes. I messed up and I accidentally used the wrong pipette, but whatever. And into their respective vessels, and then put them into those tubes. So I'm gonna go ahead and okay, so the next step is they say to add 24 drops of the glucose solution into the tube. And so here's our glucose solution. Now make sure that you're only using what you need because we're going to be using this again. So just be very conservative as far as like what you're using. The next thing they tell you to do is to observe the color of the solutions. Every single kit comes with a bunch of black construction paper. So it's kind of good to look between white and black backgrounds. And I should probably go get a white piece of paper. But you can see here, they just want to observe it before you do the test on it. Okay. I forgot to mention that when you're doing this, I would take a picture of this just to kind of remind you what it looked like before Benedict's. And if you're just watching this video, then make sure that you that you take a screen capture or something of this. The next thing they want you to do is to actually put to make your hot water. I recommend first putting in your Benedict's reagent. So they say to put 40 drops in each of the tubes. I recommend doing that prior to boiling because it's going to get cold. So, okay, I'm going to do that. In my haste to put it in the boiling water, I forgot to remind you to look at your samples before you put it into the hot water. So they should all be this kind of blue color prior to putting it in hot water. So once you have all your Benedict solution put into the tubes, put it into the boiling water, and you are supposed to incubate it for 10 minutes. So I'll come back in 10 minutes. I forgot to also mention, I'm forgetting a couple things here, is that I recommend putting your boiling water into like one of the beakers that's in there. This is let's see what this is. This is a 250 ml beaker. And this is designed to hold boiling water. So put your boiling water in there, and then you don't need to fill it all the way. I just filled it about halfway. There, it looks like it's probably under 100 mils because this displaced it. All it needs to do is really be covered by the hot water. You're going to see some pretty dramatic results. Like I'm only like a few minutes into my incubation, and you can already see some pretty dramatic results in here. So you just need to really cover it. If you put it too high, they're going to float around and potentially come out of the beaker. So just make sure that it's just covering. Unfortunately, these kids, there's no exact amounts really. And of course, if you were in the laboratory, that we would have clear procedures for you to follow. But I would just make sure that the solution is covered by boiling water. Okay, so this was 10 minutes after the incubation. It should change pretty immediately. As you can see with the distilled water, or tap water in my case, it came back negative. Do you see how it's still that nice blue color? Albumin, it's still negative. It kind of turned purple, but that's still a negative result. Here with the milk, it's interesting because it made like a gradient. Do you see that? The bottom is still blue, and then parts of it are more orange than others, but this is definitely a positive result. So this does have reducing sugars in it. And then look at our glucose. Wow, this is definitely a positive result. So what I would do is I would take a picture of this. This is the after result. So when you're doing this at home, take a picture of this and keep it, because this will be on an exam later. So what you're going to do is you're going to take these results that you got from this, and you're going to put it into the table that is in your lab write-up. So the next thing we're going to do is we are going to test for starch. We're going to wash our tubes out and then start on part 1B. Okay, so in exercise 1, part 1B, we're going to be testing for starch, and they ask us to label 4 new tubes: water, albumin, milk, and then starch. And they want us to observe what it looks like prior to adding the iodine solution. So as you can see here, everything looks pretty clear. The milk looks kind of like an opaque white. I didn't have any whole milk or whatever they said to use, cream. This is 2%, so we'll see if it works or not. They also mentioned to cut a piece of potato. So I would just slice a piece of potato or something starchy, and then just, since it comes with Petri dishes, might as well put it on the Petri dish. So that's what I did here. Okay, I do want to go over this. They said to label one of the large jumbo pipettes. I did that. So here's the iodine. Just an FYI, this is this will stain your skin and also your clothes. So just be very, very careful with this. You're only going to be adding a drop to each of your tubes and to your potato. So you don't need that much. Just make sure that you don't get any drops on yourself or your clothes. Okay, so this is the result of the iodine test, and this test for starch. You can see that the water is that negative amber color. The albumin is also that way. This is the potato. It actually took a little while for it to show a positive result. And I had a little extra iodine in the tube, and I actually put it on a cotton ball. And as you can see, that also had a positive result. So go ahead and think about that, ponder that one for me. Okay. Okay, so the next test, we're going to do a burette test, and that tests for the presence of proteins, specifically peptide bonds. So the more peptide bonds something has, the more purple this is going to become as a positive result. So what I did was I labeled 3 tubes, water, albumin, and milk, and then I added a ml to each one of them. And so the next thing I'm going to do is I'm going to add the burette reagent, and I'll be back. Okay, so I added my 3 drops of the burette reagent, and this is right after I did it, and you can already see a little bit of a positive result in one of them. So I'm going to wait 5 minutes, and then I'll be back. Okay, so these are the results. I did wait 5 minutes, but I just want to give you a heads up that with the milk, I did put another 2 drops of the burette reagent in it for it to actually turn purple. So that may be something you need to do, is put a little more in there. But you can see that if it's purple, it's a positive result. And if it's blue, it's negative. And it makes sense that the water is still that blue color. What you're going to do is you're going to fill out this table and then move on to the last exercise. Okay, so in the lipid test, you're going to be placing 1 drop of Sudan red Sudan 3 onto a piece of filter paper, and then underneath it there's some aluminum foil. Just make sure that they're about 1 inch apart, and you have to wait until the Sudan 3 is dried fully before you move on to the next step. Also, please make sure that you use a jumbo pipette to do the drops of the Sudan 3. Also, make sure that you are wearing gloves and you're wearing eye protection. Don't get this on your skin and your clothes, just like with iodine, because it will stain. Here I've put my 4 drops of Sudan 3 onto the filter paper. I'm kind of proud of myself, so I thought I'd share with you. But I'm going to wait until this dries, and then we will continue. All right, thanks. While you're waiting for your Sudan 3 to dry, go ahead and label it with what you're going to be testing on that spot. Also, I'd like to point out that my printing is pretty decent. Using an electronic pen to write on Zoom is pretty difficult. So I just wanted to show you that I do have decent printing. Okay, so I was reading ahead and I just realized that this is the only chance that you're going to have to do the lipid test. So I included more dots, and then I included the other foods that I'm going to test. So I'm also going to test peanut butter, and then also applesauce. Those are the things that were around me that was handy. Okay. While you're waiting for the results of your Sudan 3 test, which will test for the presence of lipids, I went ahead and did the brown paper bag, optional, or not optional. Is it optional? And so I did this. I went a little bit, I got a little carried away with the veggie oil, but as you can see, the lipids will go right through the paper bag. So we've got butter, peanut butter, and veggie oil. And then the ones that don't have a high lipid content is not going to go through the paper bag. So anyway, there you go. Okay, so this is the result of the Sudan test. I got a little carried away with the applesauce, so this is not a positive result. So if it is a positive result, then you're going to see that ring around the, that ring, it'll be carried outside of the original dot. So you can see that veggie oil, peanut butter, butter, even milk a little bit showed a positive result. So this is like a negative result. Like this right here, you can see that it kind of stayed within the lines. Here, this is nothing. You can see that the original dot for veggie oil, it looks like a little like fingerprint. Do you see that? And you can see that it clearly went outside of that fingerprint. It's basically carrying the dye through the paper. So it's making it like a larger dot than it was. And there's butter. You can see that that also did the same thing. Albumin, applesauce, nothing. This little quadrant here is like your negative results, and here are your positive results. And milk, a little bit, you can see that it kind of went outside the lines a little, but it's not that dramatic. Okay, so what you're going to do now is you're going to test other foods and using the same kind of tests. First, you're going to formulate some type of hypothesis. Like, what would you expect to see in a potato chip? Would you expect to see lipids? Would you expect to see carbohydrates, nucleic acids? Would you expect to see, did I see carbohydrates already? Proteins, okay? So you're going to test these other, you're going to use the same reagents, this hot mess right here. And then you're going to test the other ones. Anyway, I will let you all get to work on that, and thank you for your time and your attention, and thank you for enrolling in our Biology 112 class. Thank you. Bye.